Hi everyone. My name is Dana Stanley. I'm Associate Professor at CQ University Australia, and I'm a leader of the Microbial Genomics Cluster in Institute for Future Farming Systems in my university. So I do a lot of veterinary research and a lot of medical research in the area of intestinal health and microbial communities. I prepared this presentation just to clarify things that about 80% of people get wrong. So I'm the editor in three different really well-known journals where I handle a lot of, across those three journals, I handle a lot of and read a lot of papers in this area. I have to say that most people do not understand the difference of microbiota and microbiome, and I end up sending papers back because terminology is all wrong. So a lot of us now can get the data, but understanding the limitations of these methods is the only way you can interpret this data properly. At the end of this presentation, you will understand the difference between microbiota and microbiome. You will know what metagenomics is, and that metagenomics and microbiota is not the same. You will understand how the differences, where the differences are coming from, and you will understand how to interpret the data you get from this area and how to read papers if you are not doing this data analysis, how to read and understand the papers in this area. I intend to make this presentation very simple so that it's aimed to PhD students that want to embark into this area and are very confused with microbiota and the sequencing and the whole PCR and everything else that is involved. It is also aimed at postdocs and people that really are using this methodology in one way or the other, but do not really understand the basics of it. So let's get started. I am going to give you a presentation that I prepared for DSM Nutrition webinar, and I am just going to extend this presentation. I'm going to go slower and explain things sort of to a little bit more details so that at the end you really understand what the message I'm trying to get across. So let's go. During the last decade, we learned so many things about microbiota. So we learned that microbiota, microbial community, especially microbial community in the gut, is playing massive role in our health, and we really had no idea that this is so. So all of that was so fascinating, and it was a hot area of research. Everyone wanted a piece of microbiota cake in research space. So the knowledge rapidly accumulated. However, as I said in my introduction, there is a lot of confusion, misinterpretation, and people that do not understand the limitations, they overestimate the data and what these methodologies can provide. Two main methodologies used in this investigation, in investigating microbial communities, whether they come from gut or lungs or eye swab or whatever, or soil, for example, or, a lot of environmental microbiota investigations are really providing amazing knowledge. But wherever these samples come from, the two main ways are used to investigate the community: 16S amplicon sequencing and metagenomics. These things are fundamentally different, and a lot of people incorrectly interchange this terminology. So let's explain the basics of it. So microbiota is analysis or methodology that comes from amplicon sequencing, and it investigates taxonomy. Microbiome is one of the outputs of metagenome sequencing and investigates total DNA. So let's look at this graph. Imagine that you have microbial communities, and this Erlenmeyer flask can contain bacterial community, I don't know, from soil or from fecal material, whatever. This is one single sample. So we extract DNA, and to extract DNA, there's so many different methodologies. Again, it is important that DNA extraction process is investigated if you're doing research in this area. make sure you break the cells open properly. After that, purifying DNA using columns is not a problem, but make sure that you have lysis buffer, that you heat up the cells, or if needed, according to different protocols, but test different protocols on your samples and then see, compare the results before you finally decide that this is correct DNA extraction protocol for my samples. So after you extracted total DNA, and you have total DNA from everything that was here in the sample, there are two roads to sequencer. One of these roads is microbiota. Microbiota analysis is amplicon sequencing. It's amplicon because we're sequencing PCR amplicons. So what we do in microbiota analysis, we pick a little section of this, of one gene, so small section of one single gene, and we amplify that gene. We make millions of copies of that one little section, one little stretch of DNA from all of this. In each one of these bacterial cells, if they are bacterial, we will have, should provide amplicon for this section. So then when we get the amplicons, and say if we had, I don't know, 5,000 copies in this sample, for example, we can amplify this to 50 million copies. So what does that mean? It means that we can look deeper. It means that we can pick up, when you amplify something and multiply it to such level, you can pick up the bacteria that are present at lower levels as opposed to other methodologies. And this is why we do this amplification step, so that we can go deeper into this specific community that will be picked up by this primer set that we're using. And what we get, we sequence only these amplicons, only copies of this little section of one gene. And then when we sequence those samples, then for each sample, we can estimate taxonomy by just blasting these cells against databases for 16S sequences. This type of analysis will give you taxonomy, predicted taxonomy, not necessary gospel truth taxonomy. And I will get back to this, to species level and genus level and that type of interpretation very soon. But what we're getting here is we're looking at this one gene, and that gene, when we blast it, we're going to identify approximately which species it is, what they are, who are they, and what are they doing is a different type of analysis. In this type, we are only getting taxonomy. So, and the other way to get to sequencer is to do total DNA. Total DNA does not amplify one little portion of one gene. It sequences everything. So we fragment this DNA to smaller fragments, because obviously each sequencing machine, or not all of them, have the same length that they can sequence, but most widely used instruments at the moment is Illumina, and so we have to break it into fragments that Illumina can handle for sequencing purpose. So once this is broken into smaller pieces, everything is sequenced. So everything in this sample that has DNA, everything living has DNA. So you will pick up that DNA from every gene, not just the 16S gene, which is used in a way, this gene that we amplified in first methodology is similar to forensics investigation, where there is a gene we identify, or multiple genes we identify, not even genes, regions of DNA in human genome that can help us identify which person exactly did it when we use it for forensics. Section of DNA used for identification, for ID, personal ID. And this is what we did here in microbiota. Here we are not sequencing we're sequencing that as well, but we are sequencing everything else. So every gene in the community, we are picking up every single gene of everything living in that. There is no bias in PCRing and picking up only those communities that will respond to that PCR. So what we can get from this type of analysis is interesting, because we don't only get taxonomy, we can get taxonomy from this type of analysis, from metagenomic sequencing. We can get taxonomy, all right, but we remember when I explained before that we went, say, from 5,000 copies to 50 million copies. Amplification process will give you much deeper analysis. We can, in this type of sequencing, which is called metagenomic sequencing, we will also pick taxonomy, but not as deep as when we do PCR amplification. But then we do not have limits of PCR that I will later on explain, and I will explain this as we go through. But we also pick up genes, every gene of everything that exists in this community. When we pick up all genes, collection of all genes or all genomes in the community is called metagenome. So now do you understand why metagenome and microbiota are different things? Metagenome means collection of all genes here. When you look into metagenomic sequencing, you can get taxonomy also, which is not as deep. You can get genes. You can take those genes and put them into pathways, sort them up like, oh, this gene is for a carbohydrate metabolism, this gene is for lipid metabolism, this gene is assembly of toxin production, and so on. So we put these genes into pathways and we then look, are these pathways different? What can they do for what this community can do to host? Do they have a lot of pathogenicity related genes? Do they have a lot of starch digestion genes? Maybe they can help, this particular community can help digest completely undigestible nutrients. We can then put those pathways together and look at function and figure out how these pathways combine towards one function. Or then we can also look into collection of all genes involved in antibiotic resistance and get resistome analysis. We can look into DNA viruses because we sequenced all DNA. Virus DNA is there as well. And then we can get DNA virome analysis. And then we can use all of these little bits and pieces of a puzzle and overlap them and try to assemble genome of something that we found interesting. For example, in our recent analysis of soil samples on the side of Fitzroy River, we are finding a viral, in our virome analysis, we're finding, for example, Zika virus. And so now we want to see why we're picking Zika virus. So let's try and find the seed sequence that was identified and then try to overlap it and see, is this something, is this really Zika in Australia, or is this, for example, something similar? So we can do those things. We can assemble metagenomes of things we find interesting to also figure out what these unknown bacteria that we just assembled and kind of put that puzzle together, what do they need to grow? What are their nutritional requirements? What products they may be making? What functions they may be contributing? Are they important? Are they pathogenic? So we get so much more data. But if we do this analysis, this pathway, we get taxonomy only, but we get deep taxonomy. If we go this way, we get all of these things, but our taxonomy is more shallow. But we're not picking only bacterial amplicons, which we amplified here. We're picking up helminths, we're picking up anything else. Like if the sample was from the bird that ate a spider, we will pick up a spider genome in it. We will pick up spider sequences that will identify in 18S or whatever that, or Insectome bond library, database that is actually spider. So these two methods are very, very different. So let's go a little bit further and look into this PCR step. PCR step, this step here is crucial difference between these two methodologies. Let me just say this again. Microbiota analysis is type of analysis that is done by amplicon sequencing of 16S amplicon. This is, or maybe different amplicon, depending on what primers you take. So you can amplify bacteria, or you can amplify fungal, or you can amplify insects. They're all different primers. You can amplify one type of organisms or microorganisms in any community. That's called amplicon sequencing, because you're sequencing amplicons of one gene It's much cheaper, about 10 times cheaper than sequencing total DNA. And it gives you taxonomy only because you're sequencing part of gene that specifically because that gene can tell you who they are. Microbiome is type of analysis when you look in collection of all genomes, all genes in the community. And it's done by metagenome sequence. Meta means many, genome means genomes. So that means total DNA sequencing. Metagenomes means all genomes from community, sequencing of total DNA. And microbiome, again, is biome. It's collection of all these genes, pathways, and function. Microbiome refers to functional analysis, and amplicon sequencing refers to taxonomy. Let's move forward. PCR bias. So this is the 16S molecule. This is the 16S molecule, and 16S molecule was very misfortunately selected as a marker. You see, there is lots of different genes in bacterial genomes that are conserved, which means all species have to have. For example, in this case, this gene is part of bacterial ribosome. There is no bacteria that can exist without bacterial ribosome, right? So prokaryotes have these 2 units, obviously large and small subunit, and small 30S subunit contains this 16S ribosomal RNA gene and 2 proteins, and here it is here. So this is something that is present in every single bacteria. They all have to have ribosome. We couldn't pick a gene that is present, that's not present in all bacteria. So when you're picking up these genes, obviously you're looking at many genomes and you find something that is common for all of them. And this gene is one of the genes that is common for all bacteria, not only gene that is common for all bacteria. And that gene was selected as a marker, and everyone started using it. So now it became like everyone is sequencing 16S. All databases, all knowledge now is in 16S area. So what we are doing here now is we have these red little squares, and these are variable regions. Other regions, green regions, are conserved regions. These regions are the same in all of the bacteria, pretty much the same. But these regions are the ones where you read the sequence, and this particular sequence is the one that is distinguishing your lactobacillus from your salmonella or E. coli or whatnot. So these variable regions contain the information of what taxonomy we're looking at. But unfortunately, as you can see, there is quite a few of these regions, and this molecule is about 1,500 nucleotides. Current sequencers can do tops 500, so 1/3. Some 16S RNA could be even bigger than that, like even 2,000 nucleotides. So we're sequencing about 1/3 to maybe 1/4 to 1/3 of that molecule, a bit less. So we cannot sequence this whole molecule. We could, but it would cost a lot of money. Like we could use Sanger sequencing and get nearly all molecule, but the price would be, it wouldn't be possible to do what we're doing today. So we stuck with Illumina. Illumina can do this for relatively affordable amount now, but we have to pick the little squares. And unfortunately, we don't have consensus. We didn't all start working on the same primer section, on same primers. So someone is going to pick this region, and someone is going to start maybe from here and and go pick these 2 regions and so on. So everyone does different region. What does that mean? It means that each set of primers that you select, that, for example, primer, you put one primer here and one primer there, and you're sequencing from here to here, right? Each set of primers will pick up, none of them will pick up 100% of the community. First of all, you lost some of the community in DNA extraction, the cells that didn't open or whatnot. And now you have that DNA and you're amplifying. So each different set of primers, imagine, will take maybe, let's be optimistic and say 80% of the community. You use this set of primers, you pick up 80%, but different 80%. There could be overlap, but there is also the difference. So what I'm trying to explain, even if I use the same primers from here to here, as I used as a like imaginary example, and I just change one nucleotide in forward primer, for example, just one nucleotide, I will pick up different community. So if I move this primer, a little bit further back and further down, so obviously these conserved regions are the regions where you're going to go and look to design your primers. But I'm just giving you this as an example. So if you move this primer just a few nucleotides up or down, again you're picking up one nucleotide up or down. You're picking up different community. And this is the problem with the methodologies that use amplification. Every methodology that uses amplification, for example, this could imagine that this is insect marker gene. I have few papers where I sequenced insectome, not bacteria, for example, in community. So even in there, we have the same issue. You move primers a little bit up, a little bit down, you change one nucleotide. But in most of these other methods, we don't have variable regions. We don't, and everyone is using the same set of primers for insects. And that goes to one database, for example. And, there could be, some people can play with their own primers and whatnot, but there is more reproducibility. If you go, for example, if you want to amplify specific helminths for specific genus, someone's published primers and everyone is most likely going to go by the same set of the published primers. Here we have options, and more options, we have more variability we introduce into our data. So the other problem is that not all variable regions are created the same. Different variable region you pick, you get more or less different results, as I explained. So obviously you get one for example, difference in one nucleotide. It does make difference because species are picked at percent identity of these nucleotides. Metagenome analysis, look here, metagenome has no amplification. It does everything. So it will not miss 80% with this set of primers, 80%. It will pick up everything. There is no bias because which primers you choose, everything is sequenced. All of that 16S is sequenced. But it doesn't go as deep. So what you're going to pick up is just like when you take, when you have like a big plate of some, I don't know, ice cream, and you take just one spoon from the top, something, you will pick up bacteria, for example. If you're interested in bacteria only, you will pick up just bacteria that are most abundant, and obviously because there is no amplification, those that are most abundant will be there, depending how deep you're sequencing. Say if you go 20 million sequences, you pick up like a little scoop, but if you go 100 million sequences, you pick up maybe half a bowl. But what I'm trying to say, that usability of taxonomic analysis from this methodology is not as good. Even with PCR bias, we get more information, but we have to keep in mind that we didn't pick up everything. This is not the whole community. This is the community picked up with the primers that we selected. But you go deep then into that community and you can see things you've never seen before. The problem with these primers are multiple. This is one of the papers that I've selected by Sun and colleagues, and they're looking into the differences between different primers and whatnot. They're looking into overestimation and whatnot, and they saw that for example, V1 and V6 primers suffered the most, and V4 and V5 are okay. They're better than V1 and V6 and whatnot. There's lots of papers comparing that some primers are more prone to introduce bias. And I have a few more things to tell you that I'm sure you didn't know before. Copy number is really well known. Once we dug into 16S DNA and we started using 16S as our marker gene, we figured out that there is a problem. This gene can have multiple copies. Multiple copies in one bacteria can have more copies than one. Now, other genes that were good candidates, like RPOB or REC-A or whatnot, have one copy, and they were much better. We know that now, but back then, when we committed to 16S and started building all these big databases and everyone started publishing in 16S, people didn't really realize that this is going to be a big problem. And now we all ignore it. We all completely ignore this issue. Sun and colleagues selected, I don't know, a few thousand different cultured bacteria and evaluated how many evaluated copy number in different species. And they saw that a lot of species will have one copy. More than that will have two copies. But some species can have 15 copies, 14 copies. And this is just one group of bacteria that they investigated. Could be that there are some with, 20, 30 copies. This is a big problem. And why is this a big problem? Let's imagine this community here. We have one bacteria here and one bacteria here, but one is obviously gram-positive, one is gram-negative based on the color or whatever. We can kind of look into these bacteria. There is one here and one there. When we measure the abundance of blue and white, abundance should be the same, right? We should get 1 to 1 or 10 to 10, depending how deep we sequence based on amplicon sequences, because the number of sequences doesn't mean that this is the number of bacteria that you have in the sample. Never. It is just the number of sequences we amplified from something. So if we start from one bacteria here and one there, and we amplify them in the same process, we should get the same number. But look, we're not getting same number of sequences. It looks like there is 10 times more of this one, because this one has 10 copies of 16S genes. So this one has n equals 1, so 1 copy of 16S. This one has 10 copies of 16S gene. Obviously, we're getting 10 times more sequences from this. But there could be even worse situation. There could be even more. These copies are not necessarily always exactly the same. Let's say it has 10 copies. There are mutations. There are things that happen with bacterial genomes. And if you have one copy of a gene, you're more likely to have obviously you're going to have the same across all of the bacteria, more or less. But imagine if you have 10 copies, 15 copies. So my team, we do a lot of probiotic discovery, and we sequence a lot of individual colonies. So you pick, you isolate, I don't know, you hope it's lactobacillus or whatever probiotic strain, and then you pick a single colony, and then you purify, and then from that one species, one isolate, you pick one colony, extract DNA, you sequence, and the data for 16S comes, and you have 3 or 4 OTUs. How can that be? It should be one species. Why am I picking for single species, for single isolate, when I sequence 16S? Why am I picking 4 different species? And sometimes it can be even identified as different genus using taxonomy, because it all depends on taxonomy, right? On percent alignment and which ones were on the top of the alignment scoring system. So you have to really understand how this stuff is analyzed to interpret it. So what does that mean? This is a big problem, because it kind of tells you not just that we have 1 here and 10 here. It could be 1 here and maybe 3 different OTUs here with different number of sequences. So this and primary issues and all these issues that I started sort of touching up now are the reason that we now revising this ratio, widely cited 10-to-1 ratio. And what I mean by that, a lot of presentations, even my own presentations on microbiota, would often start with, you want to start with punchy line and get people's attention. And we could possibly say, Did you know that bacterial cells in our gut outnumber our human cells 10 times? This means that we are 90% bacteria and 10% human. A lot of people, there was a famous statement of one of the researchers that we are walking bacterial colony because we are only 10% human cells and 90% of cells of our body are bacterial. But now we're revising that. Now we're actually revising a lot of things we published maybe around 10 years ago in this area. So what it means now that we're now figuring out we way overestimated, because of all of these biases, we way overestimated the number of cells that we have in human via sequencing. So we're now kind of revising this, that it's equal in the same, as the paper says, is actually in the same order as the number of human cells. So you probably have like 1-to-1. We may be revising this again. So things that you read in papers, you have to always keep critical mind. This is like something that is very likely, but you can never take anything that you read in papers as absolute gospel truth. You have to investigate, read, and you have to understand the limitations of methodology. One other thing that I want to say is we have to pick up, and I didn't put slides for that because I could go on and on and on. I think I'm just going to explain the most important things. But let's say I have, for this sample, now I suddenly have 3 OTUs, different OTUs. I know it's one isolate. It should be one OTU. I called a lot of my colleagues. They said, yeah, we all did it. We all know it. We don't know why, but if you sequence RPO, you will just get one. So obviously then, if you want to identify, approximately identify or figure out what is most likely in your isolate, you have to actually sequence other markers and use Sanger sequencing. But let's imagine I have these 3 OTUs. I want to figure out what could be that isolate that I have, at least what genus, for example. So is it at least Lactobacillus? That would have been easier if I just did Lactobacillus PCR, then sequence the whole thing, right? But let's just see. I'm just saying, but that's actually truth. It's easier to check if you have probiotic species just by running a lot of PCRs than to send to sequencing and wait, and then you get biased results. So I pick that sequence that is most abundant. Say most of my sequences, let's say I had 1,000 sequences, round the numbers, and 500 of them were OTU1, and then the rest would be the other 2. So I picked the one, I blast N, and what I'm going to get. Sometimes you take a sequence to blast against databases and you look into alignment. So my number 1 hit could be Lactobacillus agilis. My number 2 hit could be Lactobacillus plantarum. My number 3 hit could be Lactobacillus ruminosus. But they can all be at 98% identity. All of them are at 98% identity, for example. So on an automated system, if you put the data through QIIME or the analysis, will pick the top one, the top candidate with best e-value, with best whatever p-value, whatever you set up, and percent identity. So you, in the software, you select your cutoff points. You say, I want e-value to be this, I want p-value to be that, or I want percent identity to be whatever. So those are just numbers, and they go to database and they pick the top hit. So you don't know, you look into that and you think, oh, it's Lactobacillus agilis, and you don't even know that there were 3 other different species that had same percent ID. So you can never use this methodology to say that this is Lactobacillus agilis. For all of these problems that I explained, it could be different, it could be completely different, even genus. Depends on how good is that particular primer for identifying this depends on how many different copies. If I have 3 copies, which one is it? So what I'm trying to say, if we know about all of these issues that we can see in the methodology, I am not saying that this methodology is bad or wrong. This methodology, if you understand what the problems are, you know how to interpret it. So if you look into your table, OTU table, you can say OTU most highly similar, and you put 98%, to Lactobacillus agilis appeared to be the most abundant. You cannot say, I have published papers where I used the wrong terminology in the beginning. I've been working in this area for more than 10 years now. So I don't even want to look at my old papers, because you cannot say that this is dominant when you're comparing this and this. You cannot say that the blue species is dominant over this species. You cannot, in one sample or between samples, say that this is dominant or this is not dominant. What you can say, for example, is if you have a lot of different samples and you look at one OTU, you can say that this OTU is higher here than there. But you cannot compare apples and oranges, because apple might have 10 copies and orange can have 1. So you can pick one same OTU or often same genus and say there is increase in Salmonella genus sequences or amplicons. So use the terminology. Don't make it sound like a statement there is more Salmonella than this. So you have to wrap it in a shiny paper. You have to be very careful how you word your statements. And some people put wrong statements in the paper title. Some people put wrong statements in the abstract. And when you read papers, the whole paper can refer to microbiota analysis as metagenomic. It's not metagenomic. Metagenomic is if you sequence the whole genomes, everything, the whole DNA. So it's not metagenomic. It's amplicon sequencing, 16S sequences of microbiota. And it's not microbiome because you're not looking at genes and functions. So microbiota is looking at Salmonella, E. coli, taxonomy, and microbiome is looking into, carbohydrates, metabolism, this biosynthesis of this, and so on. It's looking at functions and genes. So I hope that this is now clarified. And I don't know, maybe to save me repeating this again, perhaps watch the presentation again. When we sequence the whole genome in metagenome, first of all, we don't have amplicon, remember, but we can't get as deep into community. So you have an option to go with 16S and get the depth of one subset of community and pick up a lot of different things. But you can also do metagenomic sequencing, and metagenomic sequencing amplifies all genes, including 16S, including RPO1, including everything else. And then you can estimate which species you have, whatever, but you can also see the fungi, you can also see the helminth or insect or whatnot, or virus. But then you look into what this community can do for the host in terms of these functions. I will get into functions in a minute. But we don't have the copying, so we don't have PCR, so we don't have that issue. But we still, although we don't have PCR, we still have copy number. Different genes, other genes, not just 16S, other genes can have multiple copies. But in metagenomic sequencing, where we look at all genes, actually knowing that there is more copies of something, copy number difference between species can actually give us extra information, because some species some genes are highly variable, but in some case, multiple number of copies is associated with disease, so host state associated genes. So some genes have more copies in people that are more susceptible to become obese or to get IBD or get any other disease. And that's like genetic variation that is, for example, disease associated. And we can pick that up with metagenomic and we can investigate it properly. So it's not a problem. We also have some genes that vary in copy number as a result of environmental adaptation. For example, you can have bacterial species that, for example, has much more copies of heat shock proteins or whatever yeast species, let's say. Let's move away from bacteria and pick heat shock proteins in yeast. And it can have multiple copies and therefore become more adapted to higher temperatures or to cope with environmental stress. So yes, it could be a problem, but it could be also a benefit. Depends, again, you need to be aware of that when you're analyzing this type of data. So what do we get from microbiota? We get tables that look like that, OTU. Why OTU? Because we cannot call it species. We cannot call this species. It's not species 1, species 2, species 3. Because we had to decide about what is the similarity between the species, within the species, and difference between species. And it's variable as well. So you can have some species where difference between different isolates, different strains within the species is maybe 1% in sequence identity. So there is, out of 100%, maybe 1 out of 100 nucleotides in that, let's say, region we're looking at, there could be maybe 1 difference between different strains of this particular species. And then you can have another where there could be 5 or 6. We had to pick a number. We picked 3% difference. So if something is within 3%, all right, if something is within 3%, it's called an OTU, because that's what we think is most likely species level. Some people say no, it's not 3% difference, it's 5% difference. So we're picking up, if we go for 3% difference, we go 97% sequence identity. But what happens? We pick one sequence as a seed, and then we take all the sequences and align against that sequence. And so if there is, let's go with 100 nucleotides long. So 1 nucleotide difference, yeah, put it in this bin, it's OTU1. 1 nucleotide difference from the seed sequence, put it in. 2, 3 difference, put it in. 4 difference, no, discard. And then we fill this bucket of OTU1 with sequences, put it aside, and then we pick another seed, and then we're picking OTUs, right? Again, so yeah, so we get OTU here and OTU2. But I hope this is not difficult to understand. You have sequences compared to the seed sequence. They have 3 nucleotide difference, and these ones are 3 nucleotide difference. But the one that is 3 nucleotide difference with the seeds of OTU1 could be only 1 nucleotide difference from the seed in OTU2. So there is overlap of these buckets as well. It cannot be separated. There is not one method that does it perfectly. It's actually, there are methods now that are doing it completely different, not even using OTUs. But a lot of papers, and this is why I'm using this as an example, are still using OTUs. So basically what we get here is you have to bin them somehow. There's different methods. As I said, I'm just using OTUs. So OTU is called operational taxonomic unit because it is likely one species, but it could be that it's 2 similar species, or it could be that is 1 species but split into 3 OTUs. So 1 species could be OTU1, OTU2, OTU3. When you look at taxonomy of those 3 doesn't necessarily need to be the same. So a lot of people look at this table like it's gospel, like we have found, for example, this OTU has 88 cells in sample 1 and 650, that many more in the sample 2, and And let's say that this taxonomy says Lactobacillus agilis, and that people think that's Lactobacillus. It's not. This is just approximation based on the data. So there were a lot of papers submitted to me that I rejected. A lot of papers where people are saying we shouldn't be using PCR anymore. Sequencing is becoming cheaper. We should use 16S sequencing to identify species and infection for diagnostic purposes. Do you understand how ridiculous that is? It's completely ridiculous. We can't even know which OTU is what. We have copy number issues. We have issues with primers. We may not pick up that pathogen with our set of primers, and person is dying and we picked up something else, in the sample, and we chasing the wrong trails. We cannot call anything species level. This just came from a table, blast table as a top hit. It doesn't mean there is no other hits with exactly same sequence ID. I hope I'm not spoiling the fun with microbiota. It doesn't mean that this data is giving us idea of the number of different things, especially when we're comparing to different communities. If you look at one sample from one patient, it's ridiculous. But if you have control and treatment and you're comparing, then you can see, are these two communities very different? How much different they are? And that's where this methodology is shining. We can see, are there big shifts, smaller shifts? Is the richness higher, richness lower? But a lot of these, please never say Firmicutes bacteroides ratio. Don't use that in your papers anymore. That's such simplistic approach, and no one's using it anymore. And people that still keep citing things that we now know, and at conferences and people that work in this area, we know it's wrong and we shouldn't be doing it, but it's in papers. And I understand why people cite wrong things, because you don't know where to find information that's actually no longer relevant. Don't cite Firmicutes bacteroides ratio. Don't cite enterotypes. Don't talk about ability to identify species. Don't call this analysis metagenomic. It's not metagenomic. You didn't sequence metagenomes or many genomes or all genomes. It's amplicon analysis. Do not call it microbiome, because you're not looking at function. Call it microbiota. And do not compare apples and oranges. So don't say there is more, of this red species. If this is species level, for example, you don't say there is more red species than this green species, because red species could have 10 copies and this one could have 1. But you can say that there is slightly less of the red species, or whatever, significantly less of red species in this group than in this group, for example. Do not compare the communities using multivariate analysis. These ones overlap significantly, these ones don't. But there is more of the situations here. There is a lot of big shifts that will be picked up that should be picked up by microbiota, but they're not. And let's get into that. Before I get into the final comparison, what is functional analysis? That is metagenomic data, metagenomic sequencing. Can give you microbiome analysis, which is your functional analysis, looking into genomes and what genes they contain and what these genes can do. So if you look into that, you can detect changes in pathways related to ability of microbiota to invade host. So let's say I have two communities. One community is full of genes that encode for toxins. So I'm like, oh, this is toxic microbiota, right? This community, for example, is so enriched with genes that can degrade hard to digest polysaccharides, resistant starches, all other NSPs. So then I can say, oh, this community is very toxic in the gut of this person, and this person, for example, has a community that can degrade. This person, it could be vegan, for example, this person who can degrade very hard and complex carbohydrates. This person, third person, is got microbial community that is enriched in genes that can degrade proteins. This could be bodybuilder, right? So you get different type of information. You're not getting maybe this is this species, or you're not getting this is up and this is down. Remember how microbiota papers can sound like reading a phone book. Microbiota, blah, blah, blah. Staphylococcus was higher in this, and streptococcus was lower in that, and lactobacillus was higher in this group and then in that group. And it really is like a phone book. And that information, what do you have from that information? Clostridium is higher in group A. What can I get from it? Clostridium could be deadly, or could be short chain fatty acid producer, amazing for your health. A lot of genera have like beneficial or pathogenic is not even species. It's strain level difference. So you can have E. coli. E. coli, for example, has members of all groups from most beneficial to most pathogenic, and it's same species. So differences in, like, to get some information from taxonomy, not even species level is enough. You can't say that it's got E. coli, therefore that's pathogenic E. coli. You cannot really imply that. Even if you knew species, we can't even know species, and in some cases we can't even know genus for sure. So what do you get from microbiota is these communities are a bit different, these are more similar, there is a shift in the number, in diversity, blah, blah, whatnot. When we look into functional analysis, we're actually getting useful information. We are getting information that is definite. This person has more genes in their gut that can degrade resistant starch. We can say, oh, look, this person has this enzyme, particular enzyme is detected, the whole sequence of that enzyme, and we can actually scaffold and get the whole sequence, get a whole genome of different bacteria. So if you know how to analyze it, the whole new universe opens up. So you can get a lot more information. You can discover novel enzymes. You can pick up the enzyme. It's very similar to known enzymes, but not 100%. You can sort of assemble it and try to discover new enzymes. Then you can try to synthesize it and see what it does. So it opens the options and road to investigate the problem, rather than microbiota that gives you like a snapshot. This gives you information that is highly useful. So we can see if it produces, if it's good, what does it produce metabolites for the host that host can use, microbiota, whether it can produce beneficial or damaging or toxic, whether they can inhibit other bacteria. Are they making toxins that are meant to inhibit other bacteria? What do they make? Do we have abundance of virulence factors in one community and not in the other? So you're getting what the data you're getting, and this is not microbiota. This is microbiome analysis. If it was microbiota, it would be Staphylococcus, Streptococcus, it would be taxonomy. That's not microbiota. This is metagenomic type of sequencing, means we sequenced everything in a sample, and this is microbiome. Microbiome means we look into genes and functions, and we here picking up glycolysis genes, and we had this many copies of genes that belong to this pathway and this many here, and so on. And this is difference between these two methodologies. I hope that we now all finally understand these differences. When we use both methods, we get the best of both worlds. And this is now because microbiota is inferior to microbiome. So taxonomy analysis is inferior to function analysis. We get so much more information from function. But it's now gone so cheap, so may as well do it, right? May as well have a look at it, because it's 10 times cheaper than, and if you have money to do microbiome, may as well do microbiota as well. So the issue here is that microbiota taxonomy responds to nutritional changes. So you add more sugar to someone's diet, boom. Differences between two treatments where you just change one nutrient in feed can be massive. And you look into microbiota community in these plots and they separated and they different. And, you know, but that is not happening in things that when we look into things that should knock out microbiota, like antibiotics. We're seeing no difference. We often seeing no significant difference in things that we know are influencing a lot our gut health. So in a lot of phytogenic or antibiotic treatment, so phytogenics are products that are now replacing antibiotics in livestock industry and in veterinary science. Phytogens, your phytogen could be garlic extract, your phytogen could be oregano extract, things that can control bacteria really good. It's almost same or similar or better even than some antibiotics we used in livestock feed. But whether you look at phytogens, whether you look at antibiotic products, they were always used in low doses. They prevented diseases, they made animals grow faster, they controlled diversity. Now here comes another dilemma. We used to think that diversity, more bacteria, the better. But now when we look into, from veterinary point of view, we're actually seeing there is a lot of situations where more diversity is bad, and more and less diversity could be even, like, not too small, but you know, you don't really, having more is not always better, pretty much. So that's another paradigm that we have to shift. But let's not go into diversity. What we can see here is that big treatments like avilamycin, zinc pesitracin in small doses in the gut that cause big changes in gut morphology, in growth of birds, in their health, no disease. Microbiota, you compare microbiota, there's nothing is different. It looks like they're doing nothing. And here is one of our own papers where we looked into avilamycin and zinc pesitracin. What 2 OTUs difference, p-value nothing, like we gave them water, almost like random difference. When you look at, are these in Permanova or, you know, these multivariate analysis, are these communities different? No, really, they're not. But in all of these cases, when we look into function is completely different. You see that these antibiotics are killing toxin production, invasion of epithelial cells, all of the pathogenicity genes are targeted. So these treatments are changing function, not taxonomy. Should I say it again? These treatments, important things for animal health or human health, they are influencing functions, not taxonomy. So just think about it. How do antibiotics work? You don't have antibiotic that will wipe out species 1, species 2, species 3. You have proton pump, for example, targeting antibiotics. They target one thing, one function on bacteria. In a similar way, phytogens work. We cannot pick that up with looking into taxonomy. Taxonomy varies, but overall not much. But when you look at function, you can see this function is completely knocked out. Massive differences in function. Let me give you one example, and this is an example of oregano. Oregano given to chickens for 6 weeks, 2%, relatively high concentration, really good quality, top quality oregano, full of carvacrol and polyphenols. So we look into microbiota, nothing. PhD student was pulling his hair. It said, I'm not going to get my PhD. Oregano does nothing. Well, birds are growing better. So let's see what's going on. So when we look into functional analysis, though, then we see completely different story. What we're seeing is that a lot of cancers, genes involved in cancer, in microbiota of birds fed 2% oregano. Can you hear the way I'm saying it? You have to be very specific. So it's not that birds had less cancer. This function is talking about what is in bacteria in the gut of the bird. Bacterial community that was under 2% oregano for 6 months had less genes involved in bladder cancer, in prostate cancer, in different infections, less ability to assemble flagella, less motility proteins. That's very similar. Most of them are flagella, but you got the others as well. Less ability to make toxins, less genes involved in bacterial secretion, most of them toxin genes, and less genes involved in invading epithelial cells. Now, this is a real difference. This is what we want. We don't want it to knock off and explode the microbial community. We want these targeted functional differences. And this is why function is important. Fatty acid metabolism, okay, it came at the cost, yeah. It controls pathogens, but it reduces digestibility of fatty acids and carbohydrates, reduces steroid hormone biosynthesis, and reduces bile secretion based on function. Now, this is what is happening in genes in bacterial communities in the gut of these chickens that we sampled and did microbiota, no difference. Function, yes, difference. And then we did RNA-seq in ileum and liver of these birds. So now this was looking into microbiota communities in bacteria. RNA-seq, we're sequencing ileum of the bird and liver of the bird, and we're sequencing expressed genes, transcriptomics by RNA-seq, and we picked up all of these things. So we picked up every function picked up by microbiome functional analysis. We picked them all up that oregano does the same thing to the bird as it does to bacteria in the bird's gut. In the bird's gut, it reduced, for example, abundance of genes involved in fatty acid metabolism or carbohydrate digestion or steroid synthesis. It did exactly the same thing in ileal cells and in liver cells. It reduced genes in microbiota that are involved in bladder cancer or prostate cancer. It did the same in the person, or in this case in chicken. And this is graphical abstract of paper we submitted on liver. You can see liver gene expression by from the same birds sampled RNA-seq. And what we found, massive reduction of all estrogens, testosterone, literally reduction of steroid hormones production, which we picked up in this previous analysis. Reduction of carbohydrate and fat metabolism, inhibition of cancer categories, the same thing. The only thing we didn't pick up in RNA-seq are things that are bacteria-specific, things like ability to invade epithelial cells, where we seek, when we do RNA-seq of epithelial cells, we're not going to find genes involved into invading themselves, So everything else was picked up. So the same thing that the treatment did to bacterial cells, it did very much the same functions. Same functions were changed in looking into microbiome analysis and looking into what's happening to the host. So you get such amazing, interesting discoveries but you need to know how to understand it. So what we can also do, we can monitor. In this case, I do a lot of veterinary research, and in industry we can monitor production systems and just look into specific functions that are relevant to this industry. So this is bird industry, so we're looking into different, for example, cecum, column, crop, whatever, from different flocks, and we're looking, is there more flagella, more toxins in, is it going to jump? If we see a jump of toxin producing genes in, I don't know, ileum, we know what's going to happen. We can predict things that are going to happen if we know which function, what is going on in terms of functions in different areas. We can also look at things from day one and monitor the whole system as it goes. And if we do that, for example, we can see that, for example, here are birds before they move into sheds. So when they're in little rearing facilities, and you can see that everything is all over the place. They differ in terms of bacterial toxins, and these are different sheds, different production systems. But once they settle, and these are layer birds, once they settle into the lay, all of these functions stabilize. And even though they're a different location, different food, a lot of different things. So this is finally giving us some stability, because microbiota is all over the place in all of these situations, but functions have stabilized. Lots of stuff that we can learn, and that we can also, there's an example of functional analysis. So we had no significant difference in microbiota meaning taxonomy, but big differences in functions. So, and this is example data set of control, and then birds infected with necrotic enteritis and with salmonella. And what we're finding here, that both necrotic enteritis, Clostridium perfringens infection, and salmonella, and of course necrotic enteritis was in combination with M. aurea. In both of these cases, we have birds that are infected and that are sick. But what we're seeing is that both of these pathogens that we used in infection model reduced protein export, reduced bacterial toxins, motility, ability to infect of the whole community, bacterial community. So it's like they are suppressing all other pathogens and only they are sort of thriving. This opens completely new view to, we need to revisit these infections, because all of the knowledge we had from infections were from, a lot of data was done on cell cultures with single species. Now we have to put that species, that salmonella or that Clostridium perfringens, into community and observe all of the functions and observe how it changes these functions. Completely new area. No one's done these things before, so we can get completely new insights on how they interact with other bacteria and what functions they influence, because they're almost like antibiotics. They kill and suppress other bacteria using all of these, suppressing the same functions antibiotic produce, but they're resistant to those toxins and things they produce, and so they are not affected. So they can be the only pathogens. They can be the only one invading the gut, but they will prevent invasion of others. So we don't know much about really what is happening in our gut. The other thing, interesting information you get from the same DNA that we use to do microbiome, and I have to hurry up this, I'm going over 1 hour, so I'm going to wrap it up. I got 2 more slides. So antimicrobial resistance, antibiotic resistance is a big deal in humans, in chickens, in pigs, in everywhere. From the same DNA that we blasted against known genes and known functions, we can do it again against different resistance genes databases, and we can get profile. This is one bird, one single bird. This could easily be one person, and each one of these things here is one gene, one antibiotic resistance gene, and these ones are efflux pump group. So this is just putting them into different group based on target. So this target is tetracycline. So these are all different tetracycline resistant genes, and so on. So it's easy to find a bird or a person with 200-300 different genes for antibiotic resistance. More of these antibiotic resistant genes you carry, more at risk your health will be as the time is coming and we're getting more and more issues accumulating the antibiotic resistance genes in our body or in chicken's body or pig's body. We can get from, it's like extra information. So yes, we screened for taxonomy, we picked up helminths and insects and whatnot. Then we picked up the functional analysis. Then we put the functions into pathways. We looked for enzymes, specific enzymes. We looked in specific process that's of interest to our product maybe. Oh, but we can get antibacterial resistance or resistome profile, or we can get virus profile. If we talk about veterinary science, the same thing is for humans. We can pick up predisposition. So we can have one shed with the flock, for example, of chickens or whatever, pigs, shed of pigs, that has no viruses whatsoever. We're not picking any. And then we can have another one that has dangerous DNA virus, and it has maybe 80% birds are carrying that virus. Then we know that there is danger of specific outbreak in specific shed. And there's obviously a lot more information but you're getting that as all of these things as extra. So you're getting all of that from the same DNA. And finally, the last slide, and I'm going to wrap it up here. From microbiota analysis, from 16S analysis, we are getting overview of taxonomy, of what species or what genera, how many approximately we have, and what they could be, not what they are. We cannot ever call anything at species level based on 16S, not even full 16S. To register a probiotic, you need to do 50 different analyses to register it, for example, to prove it's that species. So you need DNA hybridization, you need a profile of sugars it's using, you need metabolites it's producing, you need a lot of things. You can't just sequence one gene and know exactly. So what we get from the same DNA, we sequence total DNA from the same, that DNA we can assemble genomes, look for all sorts of different species, assemble a whole genome, investigate what that species could be doing, and we actually then know what species we have, as opposed to taxonomy where we can predict. We can look at different bacteria we have, we can look at viruses, we can detect amoeba, helminths, other things, anything that has DNA we can detect, especially if we go deep enough. If we go deep enough, we can detect everything. Like even bacteria, we can detect all of them if we have money to sequence very deep. We can pick up antibiotic resistance genes. We can look into what pathways, what functions are changed. We can target specific virulence genes. Just look for salmonella virulence genes, depending what we are interested in. We can look for specific enzymes. We can look for novel enzymes to discover things for biotechnology. And we can integrate these analyses in a way that we can, for example, ask a question. Is increase of this particular virus influencing pathways and functions? All of these things influence each other. They are not separate entities. Everything works together like one perfectly tuned machine. And this particular way of metagenomic sequencing, or sequencing of whole DNA, can give us all of this information in one go. And yes, it is much more expensive, but it is well worth it. So much from me. This is the end, I promise, no more. You hopefully learned by now the difference between these two types of sequencing, where one we sequence amplicon of 16S gene and you get taxonomy, and the other one, metagenomics, when you sequence the whole genomes, every genome, metagenomes, all many genomes in the sample at once, whole genomes, not amplicons. And then you look into function, and that functional analysis is called microbiome, not microbiota, microbiome. Biome meaning genomes, all genomes in microbial community. I hope I didn't confuse you. If I did, you have to check on my YouTube site, and I will soon release. Try to get videos. I'm making videos for my PhD students about all of the methodologies they use through their PhDs, and very soon I will post a video about all about microbiota, for example, how to analyze it, how to interpret what we learned from it, and then each methodology into more depth. And then obviously I will post other types of analyses, how to analyze different things, how to interpret the data, and hopefully more analysis on RNA-seq and using different ways to look and investigate all of this data. But that might take some time, so keep an eye on the channel, and I will post every new video on Twitter. If you follow me on Twitter, which is at Gutsy Biotech, and here it is, the team. This is the day of dissection day. After like 12 hours of dissection, everyone still looks alive. So Institute for Future Farming System, this is the website, not always perfectly up to date, but it has pretty pictures there. And then we have Gutsy Biotech, which is overall microbiota and metagenomics and academic tweeting, and then ultra poultry, which is just poultry and veterinary research data sharing. Thank you very much for staying with me for 1 hour 15 minutes, and stay tuned for more videos that might help you understand things a little bit better. Bye.